Yeah, our lab in the past many years, and also starting with my work with Brian O'Rourke in Baltimore, our focus has for a long time been the interplay of excitation, contraction coupling, and mitochondrial energetics. And over the years, I tried to make my concepts easier and easier because mitochondrial biology is quite complex. So I try to narrow it down today on the most essential information. So during a cardiac action potential, calcium enters the myocytes and activates myofilaments and therefore induces the contraction of the cell. And the contraction and also relaxation of the cell consume very large amounts of energy in the form of ATP, which then need to be efficiently replenished by oxidative phosphorylation in mitochondria. And in this context, calcium is an important player that on the one hand increases energy consumption, but at the same time through the concept of parallel activation also governs the regeneration of energy by entering mitochondria via the mitochondrial calcium uniporter. And there, its main purpose is to activate rate-limiting enzymes of the Krebs cycle, of which you will hear much more today by the last student of Hans Krebs, Heinrich Deckmeyer. And the Krebs cycle produces NADH and also FADH2, which delivers electrons to the respiratory chain, complex 1, and thereby, and this electron transfer will lead to a proton translocation, and the membrane potential and the proton motive force drives the ATPase to regenerate the ATP. But another important product of the Krebs cycle is NADPH. And NADPH's purpose is mostly to regenerate the antioxidative systems such as glutathione peroxidase or peroxyredoxine, which play an important role in detoxifying reactive oxygen species, or more specifically, hydrogen peroxide. And over the last years, we have worked on several mechanisms, how in systolic heart failure, a mismatch between energy supply and demand can induce oxidative stress. And one problem in systolic heart failure is that there is a decreased calcium release from the sarcoplasmic reticulum, and because the SR is in very tight spatial communication with mitochondria, this decreased SR calcium release and load hampers calcium uptake in the first place. And moreover, there is an increase of sodium in the cytosol in heart failure, and while calcium enters the mitochondria via the calcium uniporter, most of it leaves the mitochondria via a sodium-calcium exchanger. And when you now elevate the sodium concentration, although this rather increases the cytosolic calcium, it has a negative impact on mitochondrial calcium accumulation because it favors the export of calcium via the sodium-calcium exchanger. So the bottom line is decrease SR calcium release and an increase in sodium hamper mitochondrial calcium uptake. And a second factor which leads to a mismatch is that the mechanical workload of the heart is typically increased in heart failure, for instance, by an increase in afterload. So in heart failure, you have neuroendocrine activation, and both adrenaline but also angiotensin can lead to vasoconstriction, and this by itself imposes an afterload, leave alone the increased stiffness of the arterial system and potentially also valvular stenosis, which can impose additional afterloads. So, and an increase in afterload means the heart and the sarcomeres need to work more, and this elevates the levels of ADP and thereby demands, in theory, more electrons from NADH. So here you have now a mismatch where less calcium and more workload lead to a mismatch, which results in less NADH, which can contribute to less efficient ATP production, and especially also an oxidation of NADPH, which can lead to the emission of reactive oxygen species. And this oxidative stress plays a causal role in the development of contractile dysfunction and also cell death through necrosis or apoptosis. And one of the last stone in this story was that we looked closer at the role of the mitochondrial transhydrogenase, which is an enzyme which sits in the inner mitochondrial membrane. And here you see what I described to you before. NADH delivers electrons to the respiratory chain for the proton gradient, and the ATPase driving force. But the same proton gradient fuels the transhydrogenase reaction to produce NADPH out of NADH. Yeah? And since this is coupled to the proton motive force, it is the equilibrium of this reaction is shifted very much in favor of NADPH regeneration. Now, in this paper, we made a rather surprising observation for us that this transhydrogenase does not only serve an antioxidative role, but can also be pro-oxidative, because while under physiological workload conditions, such as during exercise, when you do a physiological exercise, you typically have the mitochondrial calcium uptake, which produces enough NADH to fuel ATP production, and thereby also a driving force for NADPH regeneration and a sufficient antioxidative capacity. However, when you have a pathological workload, the typical thing is that here you have an increase in energy demand without a match in calcium. And then the NADH is drained towards the ATP production, and this NADH oxidation reverses the reaction of the transhydrogenase, because if there is a lot of NADPH, the driving force becomes in this direction, and then NADPH is drained towards NADH and ATP production, but at the cost of the antioxidative capacity, which should normally prevent the emission of reactive oxygen species. And we discovered this because the C57 black 6 mice from Jackson, which is the most commonly used mouse strain in the world, has a mutation of the transhydrogenase, the NNT, which makes its protein completely dysfunctional and its function 0. Yeah? And so when we learned about this in the mid-2000s, we thought this must be something that predisposes to oxidative stress, because we were only aware of this forward mode, which regenerates the antioxidative capacity. So, but when we induced an increase in afterload through TAC in these black 6J mice, and we compared them to the black 6N mice, which, by the way, have an intact transhydrogenase, and which is the regular mouse strain that you obtain from Charles River, for instance, then what we found is that against our initial expectation, there was a much stronger increase in reactive oxygen species in those black 6N mice which have a functional transhydrogenase, whereas in the black 6J mice without transhydrogenase, baseline oxidative stress, which was 8-hydroxydeguanosine staining here in the nuclei, was slightly increased, but there was no further increase after aortic banding. And this was associated with a lack of systolic dysfunction, which the black 6N mice developed after 6 weeks of TAC, and also decompensated heart failure with lung congestion, etc. And the other thing we observed is that after this TAC procedure, which in this protocol we did together with a mini pump implantation, but here the black 6N mice were dying much more often than the black 6J mice. This was an observation that a little bit before we published our study was also made by Rong Tian, however, without the identification of the transhydrogenase as an underlying reason. And now to get a proof of concept that this difference has something to do with mitochondrial ROS, we looked at necrosis and found that in fact in the black 6N, but not in the black 6J mice. We found a lot of necrosis. I don't show this here. And then we use as an intervention a compound called SS31, which is a tetrapeptide that accumulates in mitochondria and binds to chydolipin and protects from ROS-induced mitochondrial dysfunction. And with this intervention, we could rescue the mortality of these black 6N mice mostly, but it had no additional benefit in the black 6J mice. So from these studies, we concluded that we discovered a novel mechanism for mitochondrial ROS formation in response to pressure overload. And in this context, there are, of course, different sources of ROS, but you remember that there was no increase in ROS based on our assays in the black 6J mice, which also means that most of the ROS are coming actually from mitochondria during pressure overload at least. And these mitochondrial ROS play a causal role in the development of heart failure. And in black 6J mice that undergo TAC, you have to consider that mitochondrial ROS are presumably heavily underestimated. So this is important when you plan experimental studies with genetically modified mouse strains. Okay, so this has been the published part, and our story on hypertrophic cardiomyopathy. I've reported it already at several meetings, so I hope I don't bore you with this. We're still battling in the last round of, hopefully last round of revisions. So yeah, but the story, we have made a lot of experiments, so let's focus on that topic now for the rest of the talk. So here, the defects start somewhere else. No? So this is a form of heart failure that often goes back to mutations of genes that encode sarcomeric proteins. And two important of them is myosin binding protein C3 and troponin T, which we examined experimentally. Another very important one is myosin heavy chain 7. And these three, of which I show here two, have in common that they increase the calcium sensitivity of the myofilaments, which means conceptually that for any given calcium concentration, you get more force production, and this holds true both during systole and also diastole. And this is the main reason why a heart that has hypertrophic cardiomyopathy typically, especially in the early phases, does not have a systolic dysfunction, but has rather a supranormal systolic function, but a compromised diastolic function, slowed relaxation. And because every contraction or relaxation actin-myosin interaction consumes ATP, by definition, the energy consumption for any given calcium is increased. So, and this is actually the idea of that at some point Thomas Eschenhagen came up with, because he and Lucie Carrier have been working on HCM for some time before, and they came up with the idea that maybe this mismatch can also, or this increased energy demand could be a reason for an energetic mismatch, which we had previously looked at in systolic heart failure. And they were right. So we got the mice from Lucie. So the concept was that if you have this mismatch, you may also have oxidation, oxidative stress, and potentially something that contributes to the generation of arrhythmias. So we started with the mice from Lucie Carrier, and they have shown before that this mouse, which has a human, a targeted knock-in of a human mutation of myosin binding protein C3, they have this cardiac hypertrophy and a typical leftward shift in the calcium-force relationship, which indicates the increased calcium affinity of the myofilaments. And we isolated the cardiac myocytes of these mice and compared them to the wild type. And we always exposed them to a typical, to a stress protocol, because we reason that the heart is never just sitting there and doing nothing, but it's normally working, especially in the mouse. And also the other thing is that there are some reports, it's not totally agreed on, but some reports suggest that patients with HCM may be getting the arrhythmias, particularly during maximal exercise. And it's actually, sometimes you have these sport events where people die on the pitch, and quite a few of these patients have hypertrophic cardiomyopathy. So to simulate this a little bit, we do a beta-adrenergic simulation as if you have during maximal exercise by washing in isoproterenol in isolated twitching cardiac myocytes. And then we, when the isoproterenol has been working for about a minute, we increase the stimulation frequency to 5 Hertz for 3 minutes. And then we step back to 0.5 Hertz. And this is a quite kind of an abrupt but still physiological workload transition. So, excuse me. Now I have to move this here somewhere. Okay. So the first results we got out of these cells were that. So here you see the sarcomere length. Somehow my mouse is very slow now. Let me see. Fix this. Okay. So we have for any given cytosolic calcium that you see down here, we have this is really slow. Let me see if I can get this back. Yeah, let me do it for this like this for now. So while the cytosolic calcium transients at baselines are unchanged, the sarcomere length is clearly shortened in the knock-in myocytes, which indicates that for any given cytosolic calcium, there is more pre-activation of the myofilaments. And this is particularly exaggerated when you now do the 5 Hertz stress protocol, where this diastolic dysfunction, which this actually means, the cells do not completely relax back to their initial length, is even exaggerated. And in a second protocol, we measured mitochondrial calcium uptake. This is now a patch clamp-based approach, which I learned and further established with Brian O'Rourke in Baltimore. So in this patch clamp-based approach, we observed that the amplitude and accumulation of mitochondrial calcium was somewhat less in the myosin binding protein C3 knock-ins, although this was in a quite conservative ANOVA analysis, not quite significant. But you see that you rather get less calcium for more sarcomere shortening. So this tells us that there is a mismatch between workload and Krebs cycle activation. And when we now measured the NADH and NADPH redox state by the autofluorescence, and how we do it is we measure in the same cells the autofluorescence of FAD, and this is what you see here is the ratio. So it's a very well reproducible indicator of the mitochondrial redox state. You see that this is already oxidized at baseline conditions in the knock-ins, and then during the stress protocol is even further oxidized. And in the second protocol, we measured reactive oxygen species accumulation with a rather dirty ROS indicator DCF. And here you see that there is an increased oxidation of DCF, especially during the workload transition when the redox state of NADPH is becoming more oxidized. So we can follow now rather less calcium, more calcium activation here leads to oxidation of NADH, NADPH, and an increase in ROS. Now, the next thing we had not expected, but what we observed and when we look closer is then when we step back to 0.5 Hertz here, the cell has more time to make stupid things, and it starts to have spontaneous beats, arrhythmias. And these arrhythmias were significantly increased in frequency in the knock-ins compared to the wild type. Now, to get a more mechanistic idea of how these arrhythmias are occurring, we, on the left-hand side, you see the last figure we saw with the knock-ins. We used a pharmacological calcium sensitizer, EMD, which increases cell shortening without changing calcium concentration. So a pharmacological model of HCM, if you will. And here we also observed that the in the presence of the calcium sensitizer EMD, there was a more substantial oxidation of DCF. And I told you that this is a rather dirty ROS indicator and not so specific for ROS and also the location. But we did the same experiment in a mouse that overexpresses catalase targeted to the mitochondria. This is a mouse we obtained from Peter Rabinovich in Washington. So with this MCAT mouse, you see that the overall increase in DCF at baseline, but also in response with EMD, was much blunted. And this tells us that the mitochondria are the primary source, and the actual radical that we're looking at is hydrogen peroxide, because this is a ROS that is eliminated by catalase. So the next question was, if H2O2 comes out of the mitochondria, how could it induce arrhythmias? And one well-established induction of or triggering of arrhythmias are spontaneous calcium releases from the sarcoplasmic reticulum. And the reason is that the renin receptors has many sulfur residues which can be oxidized, and it's well established that renin receptors are thereby redox sensitive. So what we did is we took again this MCAT overexpressor mouse, compared it to the littermate wild types, and then again performed a stress protocol with EMD plus the additional isoproterenol stress, and then counted the calcium sparks and calcium waves. And these are experiments we did still back in Hamburg together with Peter Lipp. And what you can see is that the overall amount of sparks, and in particularly the EMD-induced increase in sparks, is blunted in the MCAT, and also the induction of calcium waves, which typically lead to depolarizations of the cell membrane and can be triggers of delayed after depolarizations. So the number of calcium waves, and also here in particular in response to EMD, was much blunted in the MCAT overexpressing mice. So this tells us that mitochondrial ROS facilitate calcium waves after calcium sensitization, in this case with EMD. So at some point, midway through the project, I met Björn Knollmann at the winter meeting in Les Diables, and over the second or third beer, he told me that he was using the troponin T I79N mutation mouse for many years already. Here you see some of his earlier work from 2001, which is very prone to isoproterenol-induced arrhythmias, as you can see here, also in vivo, and which also have the typical leftward shift in the calcium force-calcium relationship, indicating increased calcium sensitivity. And he told me that he had been doing these kind of experiments where, first of all, when so these are experiments that were done in Langendorf perfused hearts, and he is pacing these hearts at different cycle lengths, and this is rather inducing a tachycardia. And he was measuring the autofluorescence of NADH, NADPH in these hearts, and he observed that these troponin T I79M mice were having an oxidation during this pacing, and he also observed that after this protocol, there were a lot of arrhythmias induced in the I79N mice. And then he told me that he used this compound CGP. I don't know where he got it from. I think from Brian or my papers or other papers. And he saw, what is CGP? Sorry. So CGP, I told you that the calcium export out of mitochondria is via the sodium calcium exchanger. And CGP 37 something inhibits the sodium calcium exchanger. So in other words, it increases the amount of calcium in the mitochondria by limiting its export. And this kind of rebalances the redox state. And what he observed is that, in fact, in the presence of CGP, he did not see any oxidation anymore. And in particularly, the arrhythmias he was inducing under control conditions in I79N were completely blunted. And that was the point when we also teamed up and said, because we were coming from the other direction, looking at this hypothesis. So I told him this would be in agreement with what we have seen. So why don't you send us your mice and we will have a look at your mice and with our system as well. So we isolated the cardiac myocytes from his I79N mice. And in fact, we also observed that in the I79N, there was an increase in ROS, and there was also an increase in arrhythmias after the stress protocol, and we could prevent these arrhythmias to most extent with this NCLX inhibitor CGP. But this is only the half of the story, because in the beginning, after we had really clear results with Lucille's mice, after the first set of experiments, Michael Kolas, who did these experiments, he came to me and said, well, the results there are not so really clear. We have a certain trend, but it's not quite significant. And meanwhile, my technician, Michelle, she said, you know, the fur colors of these mice are different. And I forgot that he had sent us mice that were on a mixed genetic background. And Michelle, she had already done a genotyping of these mice, looking for the transhydrogenase, because she remembered our project with black 6, J, and N mice. And here is the black 6, N, and J control. And you see here now that you have a mutant allele of the black 6J, which is a truncated one in the J, and a wild type here in the black 6N. And now you see that the I79N or wild type mice we were receiving were having all sorts of transhydrogenase expression. Some were homozygous for wild type, some were homozygous for the truncated version, and many of them were actually heterozygous. And so what we did is we regrouped the data that were initially unclear, and what I've shown you before is already a cleared result, where I only showed you those that have at least one functional allele of the transhydrogenase. So that means that, and when you take the I79N on a background that does not have any functional transhydrogenase, but both truncated, there was no increase in DCF oxidation, and there was no increase in arrhythmias. And this is despite there was the same I79N troponin-induced sarcomeric dysfunction. As you can see here, the typical diastolic shortening of the sarcomere length is the very same in these mice. So this mutation worked on the sarcomeric level, but it did not induce oxidative stress or arrhythmias because there is a missing of the transhydrogenase. So this told us that the HCM-specific mutations of myosin binding protein C3, or in this case troponin T, have such a high energetic demand that apparently this pulls all the electrons away from the NADH, and this seems to be sufficient to reverse the transhydrogenase reaction again, like in the TAC model, and drain NADPH, and this gives rise to reactive oxygen species, and apparently this was coupled to the occurrence of arrhythmias. Now, to test this also in vivo, this was when Vasco Sequeira, who is meanwhile here in Würzburg in my lab, but at the time he was a postdoc. After doing his PhD with Jolanda van der Velden, he was a postdoc in Björn Knollmann's lab, and he performed the first set of experiments on I79N mice in vivo, where he also injected isoproterenol, and without getting into too many details of different anesthesia protocols, but in this protocol, the typical change that we see with isoproterenol, or that Björn and Vasco have been seeing, is that this broadens the QRS complex, which makes electrical conduction slower. Yeah? This is what you see here, the vehicle pre-treated I79N compared to the non-transgenic have a clear QRS broadening. And this is something that is a typical, gives a substrate for in vivo arrhythmias if you have a broadening of the QRS complex. And then in the second group, we pre-treated these mice with the compound SS31, and we observed that there was a blunting of this QRS broadening in the I79N, while the compound had no effects in the non-transgenic mice. However, this is not an antioxidant in the mitochondria, and this can be confirmed by the observation that the oxidative stress is increased in HCM-typical mice, but SS31 did not prevent this increase in oxidative stress in the I79N based on the 8-hydroxy-deguanosine staining. And here again, together with a second set of experiments that was done by Parik Shan in Björn's lab, we had sufficient numbers of mice and then reanalyzed their genotype, and we again found that that there was a stronger QRS broadening in those mice with a functional NNT, which do have, based on our cellular measurements, more mitochondrial ROS. Whereas in those mice that do not have an NNT, you see that the overall broadening is blunted, and if you relate it to the respective non-transgenic mice, you see that there was even a significant difference between the QRS broadening in the mice with the NNT compared to those without the NNT here in the bright red. And the second observation is that the compound SS31 very efficiently and nearly completely blunted this broadening only in those mice which have an intact transhydrogenase, whereas it had no effect at all in those without the NNT. And this is a similar result as we observed in the TUC experiments before, that apparently SS31 unfolds its protection in conditions where you have increased mitochondrial ROS. Okay, so finally, we were thinking, let us induce mitochondrial ROS artificially and see what happens to arrhythmias. And we did this by using the compound mitoparaquat. Mitoparaquat is a compound that, by its coupling to a TPP moiety, which translocates many compounds as a carrier through the inner mitochondrial membrane into mitochondria, and paraquat is a redox cycler, which is able to accept electrons from complex 1, which are derived from NADH. And it can be recycled by the formation of superoxide anion radicals, which then need to be which are efficiently dismutated. SOD is a very efficient enzyme to hydrogen peroxide, which then is left to the elimination by NADPH coupled antioxidative system, which I mentioned before, glutathione peroxidase and peroxyredoxine. And what we observed with mitoparaquat, and this is actually very new experiments we did together with the mice from Dörte Kuczynski. Sorry, this must be an I, not a Y, sorry. But we observed that mitoparaquat indeed oxidizes slightly NADPH and also a slightly oxidized glutathione. This is a MitoGRX1 RhoGFP2 mouse which reports an oxidation of glutathione, which you see over here. And with the second mouse model, which we had actually done before that already, and these are mice from Letitia Roma in Hamburg, she has also a RhoGFP but ORB1 mouse, and this is a reporter for actually free H2O2. And here we also saw that there was a slight oxidation of the NADPH redox pool, and this was associated with arrhythmias. But although we induced the arrhythmias, the actual increase in H2O2 could not be seen, which tells us that presumably an oxidation of the redox state of NADPH and or glutathione may be sufficient to trigger, at least on the cellular levels, the arrhythmias. And this we then also tested in vivo. This was again done by Parik Shan and Björn Knollmann in Vanderbilt. And the first protocol was one in which we injected, or they injected, mitoparaquat, and then shortly thereafter isoproterenol. And in fact, in the non-transgenic mice, where isoproterenol alone did not induce any arrhythmias, but in the presence of mitochondrial paraquat, there was an increase in PVCs, VTs, and AV blocks, and the total arrhythmia burden was increased. And now, but this is now just inducing artificially arrhythmias in normal mice. The critical question was whether we would have something with which we can prevent arrhythmias in mice with the HCM mutation. And here we used MitoQ. And MitoQ is coenzyme Q, which is like an antioxidant, also shuttled to the mitochondria. And this treatment, however, needed to be done for a longer time, for 3 weeks. And after 3 weeks, the isopaternal injection was performed. And here you can see that compared to non-transgenic, there was an increase in arrhythmia burden in these mice. However, the co-treatment with MitoQ prevented the isopaternal induced arrhythmic burden without having any influence on the change in heart rate, for instance, or other electrical parameters. So the conclusion from all of this is that in the normal heart, we have a very well maintained balance of anti and pro-oxidative pyridine nucleotides and antioxidative capacity. And everything is in balance because calcium, by its parallel activation, activates ATP consumption, but also regeneration. And in hypertrophic cardiomyopathy, this redox balance is in imbalance because the increased calcium affinity at the myofilaments provokes an excessive consumption or hydrolysis of ATP, which drains electrons from the respiratory chain, which under normal conditions should be balanced by calcium-induced stimulation of the Krebs cycle. But because this is not sufficiently elevated in light of this increase in workload, there is a mismatch on the mitochondrial level that oxidizes the NADH pool, which to some extent can contribute to the energetic deficit, but also of the NADPH pool, which allows an elevation of hydrogen peroxide, which then triggers spontaneous calcium release events and calcium waves, which can be an explanation for the extracellular contractions we observe. And in vivo, this could be a trigger for arrhythmias, which, however, typically requires an additional substrate. And this substrate is provided to a substantial amount by an energetic deficit that is has been described also by Sabine Hucke and Björn Knollmann some years ago, where they observed that in the same I79N mouse, focal energy depletion leads to dephosphorylation of connexin 43, and thereby the conduction of electricity along the myofibrils is slowed, and this accounts for the slowing of the QRS complex. And this is a substrate for arrhythmias, and trigger and substrate together can lead to actually ventricular arrhythmias. And therefore, points of intervention here would be to prevent oxidative stress with MitoQ, or prevent the negative effect of oxidative stress on energy production. And also, you could also consider to interfere more upstream by influencing mitochondrial calcium uptake, which I do not have here, but a very recent development is, of course, a very personalized approach to actually inactivate the myosin ATPase by myosin modulators, which tune down myosin activity and thereby tune down ATP hydrolysis without having adverse effects on the vascular resistance, such as calcium antagonists would have, for instance. Okay, and a final thing that is not completely resolved is how can oxidative stress actually contribute to the energetic deficit? And this is maybe the last piece of data. You were right. Because I took so much time to explain everything, I'm already at 46 minutes, but I think this last piece I would like to share with you as well, because it's some ongoing work by Vasco Sequeira. It has been observed clinically a lot that the typical observation in patients with heart failure, and particularly with HCM, is a decrease in the phosphocreatine over ATP ratios. And the phosphocreatine system requires a functional creatine kinase system, which is localized at different compartments, at the myofilaments and mitochondria. And what Vasco actually observed already in his time before he actually came to Würzburg, when he was still in the lab of Jolanda van der Velden, and these are data from human samples of left ventricular myocardium from patients with HCM, is that when you make Western blots with creatine kinase, you see two bands, and the lower band is an oxidized one and the upper band is a reduced one. And what he observed is that there was some substantial reduction of the reduced CK, in other words, an oxidation of creatine kinase. And when this is the quantification compared to healthy donors, there is more oxidation, irrespective of the most important mutations. And this oxidation of creatine kinase correlated with a decrease in its kinase activity, which you see plotted here. So he teamed up with Eduardo Bertero in my lab, and they were trying now to reproduce these conditions in the dish by pacing isolated cardiac myocytes under similar conditions as we have been doing before, using the calcium sensitizer EMD. And by doing so, after that, they harvested these cells and measured the creatine kinase activity. And they found that calcium sensitization was able to lead to an inactivation of creatine kinase, which could be exaggerated by higher stimulation frequencies, and which could be blunted by co-incubation with CGP, which was the inhibitor of the mitochondrial sodium-calcium exchanger. And this is just a small piece of the data he has. We are currently working more on this, but it looks like the oxidative stress that is induced by the calcium sensitization, which we had in HCM samples, but which we mimicked with EMD, induced oxidative stress that leads to oxidation of the creatine kinase and thereby to its inactivation. So this could contribute to the energetic deficit and thereby also to the substrate of arrhythmias. But further work will be required to elucidate this piece in the puzzle a little bit further. So altogether, heart failure is a condition where a mismatch between calcium-induced stimulation and workload-induced oxidation of the redox pool of the Krebs cycle can lead to oxidative stress, and these RAS can, to some extent, induce contractile dysfunction and cell death, as we have shown in systolic heart failure models, but also even in the short term contribute to arrhythmias, as I've shown you in hypertrophic cardiomyopathy, and therefore could also be targets for treatments to reduce either the amount of RAS or the implications of RAS in mitochondria, or could also argue for interventions that tune down this mismatch on the sarcomeric or EC coupling level before. And with this, I'd like to thank the people in my lab, which is Michael Kohlhaas and Alexander Nickel. Much of the work on HCM, very much of this work has been done by Michael Kohlhaas. The transhydrogenase project was done primarily by Alexander Nickel. Jan Dudek joined our group. I'm very happy about that. About 2 years ago, he came from Göttingen. He's a specialist on mitochondrial protein biochemistry and has a deep interest in cardiolipin and Barth syndrome. And I mentioned the work already by Vasco Sequeira and also Eduardo Bertero, who contributed substantially to these projects, and also the rest of my team. This picture is actually already a little bit outdated, so the group has grown in the meantime. I'd also like to point out the collaboration of many collaborators over the years. This is maybe surely not a complete list over the last years, but the projects we have been doing recently on HCM, which I'm focused on today, were primarily done with Lucy Carré, Thomas Eschenhagen, and and Björn Knollmann and Sean Parrick I mentioned. So this, thank you to all those, and thank you all for listening and for having the patience to go through these molecular details. I'm happy to address questions.
Thank you, Chris, for this very dense and full of data talk. We are grateful that you shared all these unpublished results with us. It was very interesting. We already have a number of questions lined up, but if I may, I would like to ask you a couple of questions before we move on to the questions posed by the attendees. I was wondering, your data showing that if you induce reactive oxygen species formation, for example, with mitoparaquat, you can induce these arrhythmias and calcium oscillations. The question is, because I ask you this because it has been shown that low ROS concentrations can be protective and can protect the heart against dysfunction, ischemia reperfusion damage, and so on. So what do you think is the sensor, or is there a sensor that decides what is the beneficial amount of ROS and what is when is it too much to induce damage? So do you have any idea on what is actually the target that actually decides whether the effect is going to be beneficial or detrimental?
Yes, there is also some previous work that also suggests a biphasic relationship between mitochondrial ROS and I think it was in this paper the amount of calcium sparks, and they observed that low levels of ROS stimulated calcium sparks, whereas high ROS levels would reduce calcium sparks. I'm not sure if I remember all the details, but it may be related to if you have higher ROS that you even deplete the SR so that no spontaneous calcium release with calcium occurs, because ROS can also inhibit SERCA, for instance. But Nina, you were also alluding to a phenomenon where low levels of ROS can be cardioprotective, and this is a phenomenon we know very well for ischemic preconditioning. And or another concept in more general terms is mitomasis. I think the lessons in a nutshell from many of these studies is that low levels of ROS, as you mentioned, induce protective pathways of self-defense that are able to control a larger amount of ROS under more severe conditions. Now, this is the typical phenomenon where you have angina pectoris, transient ischemia reperfusion, triggers this protection, and you're right, if you limit ROS during this preconditioning, the protection is gone. So the protective effect relies on ROS. And this is a phenomenon that is not limited to ischemia reperfusion, but apparently is in many things of biology present. And so for instance, the positive effects of exercise in athletes can also be eliminated by feeding them too much vitamin C. Yeah? So you can blunt an exercise-induced improvement of muscle function with antioxidants. And so that means that therapeutically, and this may also be the reason why, for instance, vitamins and other more global interventions of antioxidation have not been proven efficient, because you may also take away some of the physiological ROS that play a signaling role in terms of protection. And I remember also because we use this MCAT mice from Peter Rabinovich, he has a paper with Gerald Dawn where he has super expressors of MCAT, so a very high amount of MCAT, and they actually do worse. And they trace this to a phenomenon that the physiological ROS in mitochondria are required as a signal for proper mitophagy. And if you if you take away this little stress, the mitophagy system is messed up, and then that has a maladaptive function then.
But so with your intervention, with MitoQ or SS31, where you basically eliminate mito-ROS. Do you see any of these detrimental effects that you were mentioning, or you just save the phenotype?
Well, the SS31 does not scavenge the ROS, so that is what I've showed in this one figure, and it's in agreement with in vitro data we have and others have shown. It's not an antioxidant. It looks like, it's not completely understood how it's working, but it seems to have a high affinity to cardiolipin. And cardiolipin is one of the molecules that, or phospholipids that become damaged by oxidative stress in mitochondria. And according to one theory that is put forward by Hazel Sato, who actually invented this or found this compound, you protect the cardiolipin from oxidative damage with this compound. And the cardiolipin is important to keep the mitochondrial inner membrane in a proper texture, because if that gets damaged, you can induce a feed-forward mechanism of ROS, because then, that is again a concept, that electrons that are flowing down the respiratory chain, if by deficient cardiolipin dissociate from each other, you have more slippage of electrons to oxygen, and therefore superoxide formation. Yeah. Mm-hmm. So long story short, so for SS31, we would not expect to take away such a the endogenous ROS, but rather work on the downstream effect of ROS. And the other with white MitoQ, we do not know at this point. So we do have data from these MitoQ treated mice in terms of arrhythmias, but we did not look for remodeling or other parameters. A little limitation of this of these compounds, MitoParaquat or MitoQ or also MitoTempo, is that because they are coupled to the TPP moiety, which is a positive charge carrier, you are able to depolarize slightly the mitochondrial membrane potential. So it's not an inert substance. If you take cells or mitochondria and you give them too much of MitoQ or other coupled agents, you may actually damage mitochondrial function, limit respiration, and induce uncoupling. So I think here it's important to hit the right dose. Or in other, I mean, the other hypothesis one could put up, but I don't know if this is well supported by data, but since you know that preconditioning, like slight uncoupling of the mitochondrial membrane, can have such a protective function, maybe part of the beneficial effects long-term of MitoQ could also be related to such an uncoupling, which preconditions the myocardium. But this is a field that
I understand it's complicated. Thank you. And I have other curiosities, but there are like more than 20 questions in the Q&A box. So I think it's time to move on to our attendees' questions. And Peter Ferdinandi is asking, Bendavia was found ineffective in the Embrace STEMI trial. Please comment.
Yes. Yeah, it was not only inefficient in Embrace STEMI, but there is also, unfortunately, a very recent trial in patients with HFrEF, in which I think it was a 2 weeks or 3 weeks treatment with Bendavia did not, this is a phase 2 clinical trial, it did not increase ejection fraction in patients with heart failure. It's the reasons, I mean, there are preclinical studies on the dog model of MI, which have shown improvements of left ventricular function with this compound. But it's again one of these examples where indeed the translation into the clinic may prove more difficult. It could be that I mean we have not in this study with with HCM, we have not been looking at remodeling aspects. So I think this first phase 2 trial on HFrEF maybe does not report the same thing. Nobody knows if it's more efficient in the long term, but of course it's a step back at this point, that that we do not see efficiency of this compound on ejection fraction in 3 weeks. The question is, does every good compound increase ejection fraction and have REF after 3 weeks? I'm not so sure in a smaller sample of patients. So, but it's unclear how this will go on, the clinical development of the compound. The compound is also tested in other clinical scenarios independent of cardiovascular diseases, and there seems to be more promising. Yeah, I cannot comment any more than these facts on this. And again, it's difficult because we saw really convincing effects in the mouse model of pressure overload, but also HCM. But in others, many other studies have looked at even more conditions in animal studies. But this is the typical difficulty we have. We need to translate it into the clinic, and maybe we have not understood the whole, the complete mechanism of this compound.
The next question is from Martin Villa-Petrov. He asked whether you looked at CAM kinase 2 oxidation as the mechanism for ryanodine receptor leak and arrhythmias.
Yes, we didn't look at oxidation because in our hands we had we had tried to look at CAM kinase oxidation, and in our hands it didn't really work very well. Maybe we didn't try hard enough or didn't have the right antibodies. But what we did do is that we, you remember the protocol we did with the MCAT mice and the calcium sparks. So we did the same study with a CAM kinase knockout mouse from Johannes Bucks. And surprisingly, the CAM kinase knockout did not prevent the calcium sparks induction, as did our MCAT overexpression, which means, so you could imagine two scenarios. If you have oxidative stress, this can have a direct effect on ryanodine receptors through direct oxidation, or it could activate CAM kinase, and CAM kinase could phosphorylate ryanodine receptors, which was what we had in mind. But this seems not to be required for this kind of calcium sparks. So it seems to be a direct effect of oxidation of the ryanodine receptors, and which is in this in vitro scenario was not mediated by CAM kinase.
Vibarani is asking, says, mitochondria produce reactive nitrogen species as well. Did you study RNS-mediated nitrosative stress also in your disease models?
Well, we had a previous work. I mean, nitrosis. So, do mitochondria produce NO? We believe no. There is some earlier work by some groups that suggest that there is a mitochondrial NO synthase, and we looked at that again. No, this was a project we did with Barbara Casadei. So we used inhibitors and knockout models of eNOS, nNOS, etc. And although we did observe an increase in NO in mitochondria under our isopaternal protocol, we could not eliminate this by blocking mitochondrial calcium uptake. And according to the old theory, the NOS in mitochondria is an nNOS type that would require calcium activation. So we believe, to cut this maybe short, that there is an nNOS in the sarcoplasmic reticulum located, which is, as we know, very closely associated with mitochondria. And when you isolate mitochondria, you may pull down some of the SR with it. So we believe the nNOS-derived NO that we see in the mitochondria stems from the SR. However, and this was the other part of the idea, there is an idea that this NO inhibits respiration, and we could reproduce this with NO. You can limit respiration because it interferes with complex 4, I believe. However, in the nNOS or eNOS knockout mice, we did not see any effects on the mitochondrial redox state, which you would have seen when you block respiration with NO completely. You would see a reduction of the redox state. So our conclusion from this paper, which is published in JPhysiol 2 or 3 years ago, is that we have no evidence for a mitochondrial NO synthase, which could be the source of nitrogen stress from the mitochondria. However, the ROS from the mitochondria, together with NO from other sources, can of course induce nitrosative stress that can lead to post-translational modifications involving nitrogen species as well. But we have not looked so much deeper into this topic. So, but I agree that, of course, oxidative radicals are often occurring in context with nitrogen radicals as well, and there is probably a balance between this, and but we haven't looked at this any deeper.
Okay, thank you. While we're at mitochondria, if I may interject, did you look at mitochondrial function in your knock-in models? So are mitochondrial functioning well, or there is just a mismatch between ATP demand and production? Right.
This was one of the first experiments we did, obviously, because there are also some reports in the literature that claim that mitochondria and HCM are dysfunctional. We looked at Lucie Carrier's myosin binding protein C3 knock-in mouse, and at an earlier and also I think at the later age, and respiratory capacity, ROS formation in isolated mitochondria was unchanged in the knock-in compared to, so
okay.
but I cannot rule out that in the longer course of the real disease there is mitochondrial dysfunction. What actually points to this is an observation from Jolanda van der Velden's group. They looked at acetate, carbon-labeled acetate uptake by PET. And with this in patients with HCM, this is a marker for oxygen consumption. And when they put this into context with the mechanical function, they have an index of mechanical efficacy. And they find that in advanced HCM patients, the oxygen consumption is decreased. Yeah, and it could be, we know from other work that oxidative stress in mitochondria can lead to nitrosylation of complex 1. And this this is a theory, may hinder respiratory capacity and could be one explanation why oxygen consumption in these patients at the later stage decreases, although they would need more respiration for energy production, because obviously their mechanical workload, especially if they advance with even on top of the sarcomeric alterations, have extracellular matrix accumulation and stiffness of the heart, should increase. You know? So this tells us that maybe at the later clinical stages of heart failure, there may indeed be mitochondrial dysfunction secondary to the alterations that are having their root at the myofilaments.
Okay.
Christoph, may I just suggest that you try and keep the answers as brief whilst as informative as possible. Otherwise, we will never get through all the questions.
Exactly. So the next many questions are from Mike Shattuck. So the answers need to be short, really. So first question, does CGP really raise matrix calcium in healthy mito? When I looked into this, I could not find any evidence for this. A paper using mito-targeted calcium sensor, not, showed no elevation of matrix calcium by CGP. It's not a big deal, but it would really help me understand our experiments if I knew the answer.
Well, in our hands, it always did. So we not only looked at mitochondrial calcium accumulation with so we use the ROD2 Indo technology, but we also consistently see an effect on the mitochondrial redox state. And this is something that you can measure without any reporters and is nicely reproducible. And that's a quite consistent finding that with CGP, we do see a reduction of the redox state, which is not a direct proof that it's due to calcium. Of course, it could be inhibiting respiration or something. But together with the data we have on mitochondrial calcium uptake in our hands, it does.
So next question.
It's not easy to get Mike to understand his experiments. Sorry, that's a big one.
It's from Kishore Pazumarti. Your NNT background dependent results are very interesting. I'm assuming that you have done MYBPC3 knock-in ROS experiments with the N background. Do you think that similar experiments with J background would show even more aggressive phenotype in terms of ROS production and arrhythmia incidence?
So the myosin binding protein C3 mice were on the black Swiss background, and they have a functional transhydrogenase. So there we did not modify the transhydrogenase. And in the I-17N mice, it was a coincidence because we had this mixed genetic background. But here it was, so the case is here that the missense of the transhydrogenase from the J background prevents oxidative stress under these workload conditions. Not sure if I addressed the question now correctly, but so we did not do any further genetic interventions knocking in or out NNT. This was actually an unbiased approach, which was coincidentally observed, and especially since it's on a mixed genetic background, you can take it as a valid, and the grouping was only done by NNT genotype. I think it's a quite solid ruling out other genetic causes for that.
Yeah, I think you showed that convincingly. Next question is from Norimichi Koitabashi. Hi, Nori. Do you think that abnormal sarcomeric proteins lead to cardiomyocyte hypertrophy and protein synthesis due to abnormal oxidative stress coupled with EC coupling?
Yeah, so the open question at this point is whether hypertrophy can also be downstream of mitochondrial loss. I would say yes, it's well possible. There is a paper there is evidence in the literature that if you scavenge ROS in HCM models, you do prevent hypertrophy and fibrosis. For instance, with NAC, even like a rather unspecific intervention. Another interesting observation is from Jeff Malcontine's lab, and they observed that HCM and DCM typical mutations induce different patterns of hypertrophy, a concentric and an eccentric hypertrophy. And they think that a key regulator in this game is ERK activation. Calcineurin is upregulated in both instances, which increases myocardial mass. And ERK, according to their data, decides whether it's a concentric or eccentric hypertrophy. And ERK is a kinase that is known to be redox sensitive as well. So maybe this could be the coupling between ROS and the mode of hypertrophy induction. But this is untested at this point.
Yeah. Monica Glatka asks, would therapeutic inhibition of NNT be beneficial in patients with HCM? Is the level of NNT elevated in hypertrophy patients? Also, in which background are the MYBPC3 knock-in mice that you used, N or J?
So the last question we addressed, that's black Swiss mice. They have an NNT. Would it be therapeutically useful? For the heart, probably yes, but we do not know for other organs. For instance, the mutation was discovered by the fact that black 6J mice are less glucose sensitive. So, and for instance, in the pancreatic islet cells, the transhydrogenase is required for the glucose insulin release coupling. And if you don't have an NNT there, then you get less insulin release. So you may protect your heart, but become diabetic. So, and the other pharmacological difficulty is that so far there are no good inhibitors of NNT, and those that we have inhibit the ATPase as well. So, so far we do not have anything in hands. But we ask ourselves, of course, whether NNT could be a genetic modifier of penetrance of disease, but we have not answered this question yet. But what I can also say is that there are families which have mutations of transhydrogenase, and they have adrenal cortical insufficiency. So there it also plays an important role. And we're doing actually a collaboration here with the endocrinologist, and we're trying to dig a little bit deeper into this mechanism.
There is yet another question related to the NNT from Kishore. could you comment on various combinations of NNT genotypes one might expect with the mixed background mice?
Well, with the mixed background mice, there are 3 different genotypes in terms of NNT. You can have either 2 functional wild-type alleles, 2 dysfunctional truncated alleles, or heterozygous, 1 functional and 1 dysfunctional. In our hands, 1 functional allele is sufficient to have the reverse mode NNT that accounts for mitochondrial loss. You need to those alleles to actually prevent this mode of action. So 50% of the activity, and we looked at that earlier, seem to be sufficient to induce this negative effect in this case.
Okay. Rio Juni is asking, how do you think mito-ROS exit mitochondria and go to the cytoplasm? Is it just by diffusion?
That's a good question. So the hydrogen peroxide, it's definitely can pass the membrane, although there may be a role for aquaporins. This is work by Jean-Luc Baligon. So they believe, or they have evidence that the aquaporins may be channels that account for the departure of, or the emission of hydrogen peroxide. But we are very sure that the H2O2 comes out, because when we do amplex red assays, you have mitochondria and you measure the H2O2 outside the mitochondria, it's coming out. Superoxide may be more difficult. First, it's efficiently dismutated to hydrogen peroxide, but there are ways that the anion radical can leave the mitochondria, for instance, via the inner membrane anion channel, or IMAC, which was a channel that was identified by Brian O'Rourke to account for the ROS-induced ROS release phenomenon, together with the permeability transition pore. It's a 2-step mechanism where first openings of IMAC can lead to propagation of ROS signals, but if in a more severe oxidative stress, the PTP eventually opens and can also make superoxide leave the mitochondria.
Next question is by John Huison. Is there any difference in NNT expression and/or enzymatic activity in control and failing hearts? If NNT plays a critical part in mitometabolism by maintaining a high NADPH-dependent GSH-GSSG ratio, and thus the control of cellular oxidative stress, why C57 Black 6N mice are more accessible to heart failure by TAC compared to the J mice? Is there any difference in cytosolic GSH-GSSG ratio between 6N and 6J mice?
Yes, thank you, very good question. Let me start with a human heart failure situation. NNT, there is a paper in VBA around 2012, 13 or so, that suggests that in human heart failure samples, the NNT activity may be decreased. We did our own experiments on human heart failure samples and non-failing controls and observed no difference in either expression or activity of NNT. We had never published that. But the other questions are all addressed in our 2015 publication in Cell Metabolism, on the transhydrogenase and NNJ story that I showed in the beginning. But to answer directly, the reverse mode of the transhydrogenase is what depletes the NADPH. So you need a functional NNT to deplete your NADPH in scenarios of increased workload. And that also, and we've shown this, oxidizes glutathione and peroxyredoxine. For instance, in a Langendorf, in a working heart model in which we increased the afterload from 80 to 120 millimeters mercury, and after 15 minutes we snap froze the hearts, and then in N mice it was more oxidized than in the J. And in the J there was no change of the redox state. So we looked at all these steps in the cascade between NADPH, glutathione, peroxyredoxine, and H2O2.
Konstantina Statopoulou, or Dina, is asking, are there changes in protein expression of the transhydrogenase or enzymes like catalase in the MYBPC and TNNT2 knock-in mice?
I don't know.
You didn't look at that, or?
As I mentioned before, when we look at, I'm sure maybe Lucy has data on that for gene arrays, but let me answer it this way. I'm a believer in in activities rather than expression levels. You know what an mRNA result can give you. And when we look at the isolated mitochondria from these hearts, myosin binding protein C3 knock-in and the wild-type controls, we measure not only respiration but also H2O2 with Ampex red and also superoxide with spin trap, which is the most the gold standard for these ROS. And we see no differences in H2O2 or superoxide in state 2 or state 3 respiration, so the whole spectrum of activation. And from that, I conclude that, I mean, if there was an increased expression of, let's say, well, glutathione peroxidase, for instance, you would probably see it there. But your point is well taken, because catalase is maybe more in the cytosol. But I don't think we looked at that specifically. Maybe we did, but at least I don't remember it.
Okay.
But good question.
You're basically more than halfway through the questions that we have, Chris. So you tell me how much more strength you have and how long you want to go on.
I have a glass of water. I can go on forever.
Okay.
You need a glass of wine, I would suggest.
Exactly.
Exactly.
That's afterwards. Mike Shattuck is asking, do you think your MitoROS might activate PKG, forming a PKG dimer and changing calcium uptake into SR via phospholamban phosphorylation, not only via increased calcium release from the ryanodine receptor?
That's very well possible. One thing we observe when we wash in mitoparaquat is that in many circumstances, actually the calcium transients initially can increase. There is all, I think there are, I mean, the effects of reactive oxygen species on EC coupling are, there are a zillion ways of affecting this. We discussed some of them already, ryanodine receptor, chem kinase. There is also data that SERCA activity can be modulated by ROS in a positive and a negative way, maybe also through this biphasic concentration dependence. And PKG, we have not looked at. I'm sure it's possible, but we never tested it directly ourselves. So I cannot properly answer this.
A question from an anonymous attendee. How do you differentiate mitochondrial versus cytosolic calcium transients? Do you use any specific dyes?
Yeah, I didn't go too much detail here. This is the work I did I started with Brian and Rook. So measuring mitochondrial calcium in adult cardiac myocytes is a challenge, this way or the other. There are a number of ways to do this. I think the most specific reporters for mitochondrial calcium are typically those that are genetically encoded. The downside of this is that if you infect myocytes with genetically encoded reporters, you may have a de-differentiation of the T-tubular system or of EC coupling in general. And because mitochondrial calcium uptake requires this calcium-induced calcium release, this could be a downside. We use a technology in which we preloaded cardiac myocytes with a calcium indicator called ROD2. It goes primarily, but not only, to mitochondria. And to control for the cytosolic contamination, we patch clamp the myocytes and wash out the ROD2 traces from the cytosol with a patch pipette solution, but at the same time bring in Indo as a salt that stays strictly in the cytosolic compartment. And we established this in our paper in 2006, and we did a lot of control experiments. So for instance, we included Ru360, which is a quite specific inhibitor of the MCU, calcium uniporter, and with this we can selectively decrease the mitochondrial calcium transient and at the same time slightly increase the cytosolic calcium transient. So we were very confident that with this technology, we mostly report mitochondrial calcium. What is still a debated issue is how much calcium enters the mitochondria. I think it's quite clear that it can enter it quickly, but if it's a beat-to-beat regulation and the amounts that get in per beat is still not completely resolved. Let's put it that way.
Another anonymous question is from an anonymous person.
Although most data point to the fact that the overall amounts that enter the mitochondria on a beat-to-beat basis are rather low and presumably inconsequential for EC coupling. So the main thing is the accumulation of calcium that dictates the actual steady state level in the mitochondria that account for Krebs cycle stimulation. Sorry, Nina, to interrupt.
Well, so another question is the data of increased arrhythmia in mitoparaquat treated non-transgenic mice done in vivo or in Langendorf?
Yeah, so first I showed you cell data, that's clear, and the others were done in vivo. So it was living mice injected mitoparaquat. Actually, Björn and Sean did two protocols, one with a low concentration of mitoparaquat, which required additional isoproterenol stimulation to induce the arrhythmias. And they had one protocol where they injected a higher dose of mitoparaquat acutely, and that by itself was sufficient to trigger arrhythmias, even without isoproterenol. But it's probably a toxic dose.
Danuta Cordery is asking, do you think that transgenic mice on J background, like Bjorn's TNT I79N, original mice will not properly project any energetic changes that may occur in oxygen production consumption due to sarcomeric hypertrophic cardiomyopathy mutation?
Yeah, well, so yeah, I mean, what I can say is the result that I've shown that by retrospective analysis of the NNT genotype, we observed that the missense of any functional transhydrogenase apparently led to a less severe broadening of the QRS complex in this lung. This was actually in this in vivo situation. We did not look at many more things. We actually, but this is, so we assume based on this observation and the cellular observation that the missense of transhydrogenase may reduce NADPH oxidation with its consequences, which may be more H2O2 and which may be oxidation of the glutathione redox pool, but we didn't look at all at the cascades in that NNT dependent manner in this paper, because we did hundreds of experiments with this NNT involvement in the TAC experiments, so we did not redo all the cascades again, but the data so far fit together.
Dunja Aksentievich would like to know, in your experiments where you referred to energetic stress impaired energy production, which metabolic substrates were used, glucose alone or combined with other metabolic substrates?
In which experiments? In the cell experiments?
So she says energetic stress impaired energy production, but I believe you pointed out that there was no energy impairment. It's a mismatch, but mitochondrial were functional. So I guess she's referring to the mitochondrial substrates.
Yeah, so I can tell you that in the cell experiments, we usually take luxurious energetic substrates by adding glucose and pyruvate. In the isolated mitochondria, we use I think the Langendorf, I would assume, was done in only glucose by Björn, I would assume. And in the isolated mitochondria, we used pyruvate malate as substrates for respiration to look at mitochondrial function.
Okay. From Luc Bertrand, glucose and fatty acids or even other alternative energy providing substrates differentially enter into ATP production. Do you know if your paradigm is differentially affected by these different metabolites?
That's a really good question. we do not know. The cell experiments certainly were only done in glucose. So this is a systemic error we make in all our cell experiments. We always use glucose. I mean, everybody who uses fatty acids in cells knows that this is a disaster. But the good news is that when we do it in vivo, the concept still stands. So the mitoparaquat or mitoq interventions in vivo showed efficiency in both directions. So I would assume that the mode of action that we're looking at is independent whether glucose or fatty acid are the primary upstream substrate for Krebs cycle.
Rio Juni says that the penetrance of HCM mutation is incomplete. It seems there is a second hit, in addition to sarcomeric protein mutation, that contributes to the manifestation of the disease. What is your opinion on that?
I fully agree. I mean, this is not something that is a matter of a matter that needs to be resolved for HCM, but also DCM, of course. The fact that you can have patients which are genotype positive but have no phenotype clearly calls into question that there must be modifiers. And whether this is other genetic modifiers and or environmental stresses such as high blood pressure, diabetes, pregnancy. So for instance, we learned that not only in DCM, this is now not HCM, but titin mutations are common in DCM patients. And if you then look at PPCM, so peripartum cardiomyopathy, or doxorubicin-induced heart failure, those who actually get this disease also are more frequent carriers of mutations in titin. So this could also mean that in an HCM scenario, a genetic predisposition plus a second hit mediates the penetrance of the disease. And to identify these triggers is, I think, the task that we are facing now to better, I mean, when you identify one patient with, let's say, HCM, you want to do something good for the relatives, the brothers, sisters, and maybe children of these patients. So it would be good to find out what these triggers are and whether we can prevent the penetrance of the disease by dedicated interventions.
Julia Liu would like to know, would you predict that in conditions of energetic mismatch, that increasing mitochondrial calcium uptake by enhancing MCU activity, pharmacologically targeting the complex or mutations in the subunits, would have positive effects? Or would NCLX inhibition by CGP also be required? At what point would MPTP opening become a concern?
Mm-hmm. You already give the answer yourself, because it's a two-edged sword. Biology is often the balance of something in the middle. If you have too much of one thing or not enough, both is normally not good. So we believe that, and this is actually a controversy out there on is there too much calcium in mitochondria, even in, I mean, I think there is general consensus, not complete consensus, that in ischemia reperfusion, acute ischemia reperfusion, you rather get mitochondrial calcium overload that activates the PTP. In this scenario, inhibiting this calcium uptake would be efficient, yeah, or is efficient. Although there are also surprising results with the MCU knockout mice. In our scenario, what I told you about workload mismatch, we would of course be in favor of rather increasing mitochondrial calcium uptake. There is, I mean, you're probably aware of SGLT2 inhibitors, that they have this surprising effect on, beneficial effect on heart failure. And the mode of action is not completely clear. What seems to be clear is that it does not have much to do with glucose, because the non-diabetics benefit from this compound the same way as the diabetic patients when they have systolic heart failure. And Kurt Surbier and Ton Barcher observed that SGLT2 inhibitors can inhibit the sodium proton exchanger in cardiac myocytes and lower cytosolic sodium. And they also showed that with these compounds, you increase mitochondrial calcium. So maybe some of the beneficial effects of SGLT2 inhibitors could be explained by increasing mitochondrial calcium uptake in systolic heart failure. But this is not completely resolved. Of course, we have a strong opinion that you should rather increase mitochondrial calcium uptake due to this mismatch. But if you then run into, if you take a drug to increase it and then you run into MI, it could be a disadvantage if you then have too much calcium uptake.
Definitely a disadvantage. Last two questions. So the one before last is from Marisol Ruiz Meana. She's asking, do you think that reversion of transhydrogenase could be relevant in other pathological contexts like ischemia reperfusion injury contributing to oxidative damage?
That's a good question. So Rainer Schulz made experiments with our NNJ mice. Well, NNJ mice, you get them at every corner, but we asked him to do ischemia reperfusion and measure infarct size. So this was acute MI, ischemia reperfusion, and there was no difference in infarct size related to area at risk in NNJ mice. That's the good news for anybody who's doing acute MI experiments on J backgrounds or so. But I would not exclude, just based on this experiment, that it does not play a role. We could talk for hours how the equilibria change in the mitochondria and how this, you could have reasons for NNT going forward and reverse in different phases of ischemia reperfusion as well. Other scenarios of reverse mode NNT, except for, so I think it's something that is particularly specific for very energy consuming organs. Because obviously if you drain ATP, that is a situation where you deplete NADH and you could reverse the reaction against this proton motive force, which is a very strong way of holding it in the forward mode. And that is also the reason why in most tissues that are not so much energy consuming, the dominant direction of the transhydrogenase is the forward mode, and such as the pancreatic islets, etc. So and the other interesting thing is that we observed, that's also in the 2015 paper, that if you take a brain or the skeletal muscle and measure NNT activity or expression, it's very, very low. So the NNT is particularly present in the heart and not so much in other tissues. Other energy consuming tissues such as the skeletal muscle and the brain. So it has many phases. The other thing that is remarkable is that there was a recent Nature paper that actually resolved the structure of the NNT in the reverse mode. So I'm happy that also other people than just myself are talking about the reverse mode NNT. And it's even in Nature, in contrast to our paper.
And so the last very interesting question is from Matthias Merikse. He says that mitochondria-derived ROS are supposed to be an important driving mechanism in aging. So the question is, are there differences in cardiovascular aging phenotype between J and N mice?
Not that I'm aware of. Both are. So J mice live a normal, happy, long life. The first MCAT overexpression model that was published in Science by Peter Rabinovich was on a J background. And in that mouse, mitochondrial catalase is still extended lifespan. So it doesn't mean that, so, what was the question?
Whether the cardiovascular aging phenotype between J and N mice is different.
Yeah.
Because of the mitochondrial ROS that are involved in aging. Has anyone even looked at that? Whether they live longer, whether hearts are differently remodeled during aging?
I'm thinking if we looked at it, but I don't remember. So I don't know, Matthias. I don't know completely, but it's a very good point.
It's the next experiment.
Exactly.
Aging. Oh, let's look at aging. Years of experiments. Well, you could probably buy 2-year-old mice from Jackson and look at them right away, right?
Yeah. You can, maybe you don't believe it, but you've answered all the questions. So thank you very much, and you deserve a glass of wine now.
Large glass of wine.
Well, thank you for this nice seminar. The questions have been outstanding, and thank you for your patience with this crazy complex data. And it was a big pleasure for me this evening. So thank you.
Thank you very much for your patience and for sharing the data with us. And thank you for all the attendees that have stayed out so late. And we will see you tomorrow for Dr. Tegmeyer lecture. Very important. So please be a lot of it now, numerous. Thank you very much to everybody. Have a good night.
I'd just like to thank Christoph, by the way, if you don't mind, for exceptional science, even though you're terrible at sticking at time, but exceptional science and really, really quite profound answers to questions. And you're actually close to the record in terms of time as well. So for a webinar. No, but this is a good thing. I think the richness of these webinars often lies in Q&As. And I think you've demonstrated that today. So thank you once again. And Nina, thank you for putting up with us as well.
No, it was a pleasure.
Thank you. All right.
Bye.
Have a nice evening or afternoon, everyone. Bye.
Yeah.
See you tomorrow.
Bye.